Thank you, Dennis. Thank you, everyone in here. Thank you for giving me this opportunity to talk about one of my research projects. So the title of my talk is Modeling Residue Pattern of Peptide Recognition Domain in Protein Sequence using Stacked Generalization. So I'll start with a little bit of overview. Protein-protein interactions plays a key role in the biological as well as pathogenic processes in cell through physical interactions among multiple proteins within a complex. And it is known that about 40% of the PPIs involve recognition of linear peptides by globular receptors that can induce peptide binding. Human proteome contains millions of peptide motifs that are typically part of disordered regions and bind with appropriate partners through transient interactions. Approximately 22% of human disease mutations in disordered segments of proteins occur in peptide motifs. So with that background observation, what we did is in March 2016 that we cross-checked each of the 694 sequences from this broad database, which is a database of intrinsically disordered protein with 300,000 protein sequences from Protein Data Bank. And what we found is 22% of IDPs or proteins with IDRs, which is intrinsically disordered regions of this broad have structures in Protein Data Bank. Some of the IDRs or IDPs have multiple structures in PDBs for exactly the same sequence, and the largest cluster we had is 140 structures for one single sequence. So with these observations, the problem that we wanted to attempt can be defined in a coarse-grained way as given two proteins, one with a peptide recognition domain such as histocompatibility protein and another protein which is a peptide, identify if they interact or not. However, this problem can also be defined in detail that given two proteins identify the residues or regions of interaction. So here is the visual illustration of that problem that here the major histocompatibility protein is basically interacting with a peptide, and these yellow residues are actually the residues of interaction. So under this project, our target was to characterize these peptide recognizing residues. So our work steps were we proposed a new computational tool to predict peptide binding residues of proteins from sequence alone. A set of protein complexes with wide range of peptide recognition domain was collected from PDB and were annotated. Using a set of sequence-based features, we developed a staked machine learning model. The final model was found promising in identifying peptide recognition domain prediction. Identifying peptide recognition regions or residues. So here's the first step of our task, which is the data collection, and we did the data collection from Protein Data Bank using the following criteria, and I'll not explain a little bit detail, but I'll be happy to answer any question later. So in our data set, we had a wide range of peptide recognition domains, such as major histocompatibility domains that usually interacts with peptides of length 8 to 12. PDZ domain binds with C-terminal peptide, SRC domain, SRC2 and 3 domain binds with different types of motifs and actually phosphopeptides, and 14-3 domain, WW domain, and etc. And importantly, we also had enzyme inhibitor complex and antibody antigen fold with Fab complex in our data set. Then we annotated the peptide binding residues or region based on the 3D coordinates of the residues in atomic level, specifically the atom records in the PDB file. So what we did is we annotated an amino acid as interacting, which is this yellow highlighted residues and the orange highlighted residues here, if at least one of its heavy atoms stays within 6 angstrom distance from a heavy atom of a peptide, which is highlighted in pink here. On top of this actual annotation, what I call as actual annotation, we did a smoothing to have a local region of interaction, like a beta sheet here and a helical region here. Then in the next step, we did the feature-based encoding of the protein residues to characterize the peptide binding residues. And as the features, we have used amino acid profile, chemical properties of the residues, evolutionary profile to indicate conservation information, local backbone structural profiles such as the secondary structure properties, backbone angle values, and accessible surface area values. We have also used the flexibility profiles such as disorder probability and backbone angle fluctuations. We used energy profile, which is the sequence-based estimation of energy per residue, and we have also used terminal profile, which indicates if a residue is in the N or C terminal of the proteins. So here is the machine learning part where we used model staking. So model staking is a powerful ensemble learning technique. It's a two-tier learning. First tier involves base learners, and the second tier is the meta learner step. Underlying idea is different complementary base learners can incorrectly learn different regions of feature space. So a meta learner can be applied on top of that to correct the improper training of the first tier. So here I'll be explaining a little bit of training and testing of the base learners and meta learners. So the train and test step of base learners are like pretty straightforward, which is like you give the full training set into the base classifiers using the features, and then you have your base models. You feed your test set, and then you have your outputs here. However, the train and test of meta learner is not that straightforward. Here, what we did is we split our training set into two different folds. And then we fed the first fold into the base classifiers, and we have one set of base models. And we fed fold 2 to predict, and we have the output probabilities of fold 2. And then the next step, what we do is we feed fold 2 as the training set, and then we have another set of base models, and then we feed fold 2 to predict, fold 1 to predict, and then we have the fold 1 probabilities. So here we have the full training set probabilities, and then we feed these probability values into the meta classifier, and then we have our final model. So these are like the key step of the staking method, which uses independent probability outputs from the base step to train the meta step. In this classification task, we have used 6 different base learners, such as support vector machine, random decision forest, access tree classifier, gradient boosting classifier, k-nearest neighbor, and bagging classifier. As the meta learner, we use logistic regression to non-linearly combine the output of the base learners. Excuse me. Here I have shown the performance of the base learner. So the highlight is the optimized SVM was actually found the best base learners for this classification task. However, the key part of the staking model is to select a set of base learners or base classifiers that are really uncorrelated with each other. So SVM, GBC, and KNN were found to be uncorrelated predictor for this application. So what you can see here is the Pearson correlation coefficient of the outputs given by SVM, GBC, and KNN that were found a little lower. So we selected these 3 classifiers as the base learners. So here is the final model that we call Peptide Binding Residue Predictor, or PBR Predict, where we applied SVM, GBC, and KNN is the base step, and logreg as the meta learner. So here I acknowledge the tools that I have used for this classification modeling, and for the interactive visualization, I have used PyMol. Here's the comparison between our model PBR Predict and the only one existing method out there at that time, which is called Sprint. So a comparison gave us interesting results, such as Sprint can really result high true positive rate, but at the cost of very high false positive rate. But if you see the balanced measure for the binary classification evaluation, for an example, Matthews correlation coefficient or F1 score, then our method was far better. So this comparison gave us an initial idea that Sprint may be suffering from overprediction. To demonstrate it clearly, I will show you some case studies now. So here's the case study that I did with major histocompatibility protein. So in this plot, you can see the red highlighted residues. These are the actual peptide binding residues that interacts with this cyan peptide here. And here's the prediction output of our model, which is like quite near perfect. And then here's the output of Sprint, where you can see that it is annotating the residues at peptide binding residues, which are the pink colored residues that are like far distant from where actually the peptide is. As the second case study, I will show you a protein complex with a protein with polar box domain and a peptide which is PL55. Here is the peptide in cyan color, and here is the protein with polar box domain, and here is the actual peptide binding residues in red. Again, here, and this is the output of PBR Predict, which is our model, and here is the output of Sprint, and you can see that it is again over predicting peptide binding residues like almost everywhere. And here is the last case study that I will show, which is a complex between a protein with 14-3-3 domain and a peptide with phosphoserine. And the red part is the actual peptide binding residues, and the yellow is our prediction. And the pink one is the sprint annotation. And here also you can see that this is the peptide, and it is highlighting these residues that are far away as peptide binding residues. So with that, I have come almost to the end of my talk here. I just want to highlight that the extended part of this research project has been recently accepted in Oxford Bioinformatics. And in this paper, you will find like a lot more case studies and proof that this model was like quite exhaustively evaluated, and it is a robust model. And the software is out there available to be installed in a Linux server, and it's an open source. So with that, I would like to thank you, my past laboratory, which is our Bioinformatics and Machine Learning Lab at Computer Science Department of University of New Orleans. And I would also like to thank my current institution and my mentor, Dennis, for allowing me to continue this work even after my PhD. Thank you very much.